Thank you, Dan. Thank you all for being here. It's almost amazing to see how much we can still learn from studying DNA tumor viruses. Before I begin, these are my disclosures. I've received some research funding from AstraZeneca. And so for the purpose of this talk today, I wanted to discuss some of our interests, longstanding interests in understanding the mechanisms of metastasis. And in the recent couple of years, we've been focusing on the problem of brain metastasis or metastasis to the central nervous system. And here's the clinical challenge. So brain metastasis can arise from multiple types of tumors, predominantly lung, breast, and melanoma. And as these patients are responding to systemic therapy, but eventually relapsing, the incidence of CNS relapses is predicted to increase over the next couple of years. And as one of my good close colleagues, Veronica Chang always tells me, and I always listen to her when she tells me anything, it's actually been very difficult to study brain metastasis because although the incidence is relatively high, very few of these patients are actually, less than 10% undergo surgical resections, and therefore obtaining tissue to study this disease is difficult. From a biological standpoint, I think what's fascinating me is encapsulated here by these two images taken from the clinical literature, where you can see that in addition to originating from different types of cancers, brain mets can also manifest or present in different heterogeneous patterns of disease progression. So for instance, some tumors are well circumscribed and focal, where there's other brain mets appear as multifocal disease with various patterns of invasion in the brain parenchyma. And so what are the mechanisms that drive this? Are they genetic? And if they are these mutations shared with the private tumors or unique to the brain metastasis? Can they be epigenetic? And if so, are these epigenetic changes intrinsic to the cell types, tumor cell types, or are they induced by the tumor microenvironment? And finally, in the case of relapse, this is due to poor penetration of systemic drugs or other factors from the microenvironment. And so the approaches that we use to study brain metastasis are really 2 to 3-fold. The first is that in collaboration with Veronica Chang, Abhi Patel, and several others involved in lung cancer spore, we're leveraging cerebrospinal fluid biopsies from patients to identify drivers of CNS metastasis. This is a new initiative. I won't have time to discuss this today. What I'd like to focus on today is mostly preclinical approaches to identifying adaptive mechanisms of brain metastasis outgrowth and drug resistance using preclinical models. Another major thrust in my lab is we're interested in the relationship between developmental pathways and how these drive tumor progression, particularly in lung cancer. And I'll only touch upon this topic insofar as it is relevant to the discussion of brain metastasis. So in my lab, we use several different models. I like to tell all my trainees that when they come into the lab that we're not model Nazis, we're equal opportunists, recognizing both the advantages and limitations of each of these models. Xenografts, patient-derived xenografts, and also syngeneic models. One of the approaches I do want to mention in a bit more detail is to study metastasis is, while it's easy to document, or relatively easy to document metastasis, it's another thing to actually quantify these events. And the approach that we use here is that we can take tumor cells, label them with different reporters, and inject them into circulation of mice. And what this allows us to do is quantify, document and quantify with some accuracy some of the very early steps of metastasis, which include breaching of the blood-brain barrier, co-option of the CNS blood vasculature, latency of these disseminated tumor cells as micromets that persist for some time before they start to grow out as macrometastasis. And I just want to mention that experimentally, and we believe that this happens in humans as well, most of the attrition of tumor cells, or the bottleneck, as you will, occurs during these early steps. And these early steps are not radiologically detectable. And in fact, it's not till later in these model systems that you can start to see large macrometastases and by MRI, as you might in humans. And this is done in collaboration with Famin Haider and his former postdoc, Maxime Parent. So really, the challenge here is how do we identify adaptive mechanisms at these early stages, which we would argue are rate limiting for brain metastasis. So in my lab, we're also interested in optimizing or leveraging several types of transcriptomic approaches. And we use we had a really simple idea, which goes as follows. Can we leverage recent advances in our ability to map transcripts of low abundance in a species-specific manner in tissues? And secondly, if we apply this approach to xenograft models, could we differentiate or discern gene expression changes that are coming from the tumor cells? Because these would be human genes that are coming from the human tumor cells that are then transplanted in the murine hosts, and therefore the stroma will be represented by changes in murine genes. And so this is a proof of principle experiment which take a well-characterized lung cancer cell line that metastasizes to the brain. We grow it in culture, grow it as a flank tumor, which is something that people commonly do to study tumorigenesis. And then we allow these cells to also disseminate and form brain metastasis. And then what you see here, this is just based on H&E, looking at some of these, what the tumors look like. If you have a flank tumor, the tumors are quite nodular, mostly tumors with some stromal, high grade. But if you inject the same tumor cells and allow them to form brain mets, you can see that they form these multifocal, diffusely invasive lesions in the brain, highly reproducibly. And I would be hard-pressed to ask anyone in my lab to try to microdissect these lesions. And so clearly the microenvironment matters. And so when we apply our pipeline, which we call brain met xenograft RNA-seq or brain met sequencing pipeline, what do we see? And in this case, because we were somewhat lazy, we just decided to take the whole brain tissue more or less and see whether we could grind it up and submit it to RNA sequencing and apply this pipeline. And the data is actually quite informative, even in this simple-minded experimental design. So here's just the mapping proportion of the transcripts to either the human genome or the murine genome in subcutaneous tumors. And then here's the same thing in brain mets. And you can immediately see that most of the transcripts in the subcutaneous tumors are as predicted, mapping to the human genome because there's higher tumor content. And then the opposite is seen here, where in the brain mets, most of the transcripts that are mapped here are coming from the murine genome, and 6% are coming from the tumor genome, presumably from these diffusely invasive tumor cells. Despite this heterogeneous pattern of seeding, we can get very clean data. So here's looking at tumor gene expression profile of tumor cells that are grown in culture, either grown in subcutaneous tumors or the same tumor cells grown in brain metastasis. And just parenthetically, in some of our models, we observed this very reproducible pattern of some tumor cells colonizing the forebrain versus the hindbrain. And so what Emily Wingrove did in the lab was basically cut these two pieces and do gene expression profile on them also. And while the tumor cell gene expression profiles between these two regions is very similar, you can distinguish or find a subset of about 100 or 200 genes that distinguishes tumor cells that are growing in the forebrain versus the hindbrain. And then finally, in the same samples, we can look at the stroma. And again, you can see that depending on where the tumors are growing, we can find very specific context-dependent gene expression signatures. So we think this approach is fairly useful, and especially if we consider that we're trying here to capture things that are happening in situ at some of the earliest stages of brain metastasis and in different regions of the brain. So another area where this approach might be very useful is to study mechanisms of drug resistance. And in this case, my lab has been particularly interested in studying lung cancer. And as many of you know, a relevant subtype of lung cancer are patients that have EGFR mutations. About half of these patients go on to develop brain metastasis. And again, as most of you will know, the clinical landscape for these patients has changed significantly in the recent years, where by now, with the development of third-generation tyrosine kinase inhibitors that specifically target these EGFR mutations, and this is quickly becoming a new standard of frontline treatment. And this is a sub-analysis of the FLORA trial where they examine Osimertinib, third-gen TKI compared to first-line TKIs. And you can see that in terms of sub-analysis of this trial shows that Osimertinib has superior control of CNS disease progression. You'll have to take my word for it. We've collaborated with AstraZeneca and showed that part of this efficacy we think is due to the fact that Osimertinib is highly brain penetrant. But despite this penetrance into the brain, which has been historically considered a sanctuary site, we can obviously these patients still develop TKI-resistant disease, and we can model this. So in here, you're looking at tumors that are growing in the brain that we're quantifying here. These animals are all under treatment. We can have tumors that grow preferentially in the brain under continuous treatment of Osimertinib. These tumors, they can be compared here to tumors that share a similar lineage. I won't get into the details of how these were generated, but essentially these tumors can be compared to one another. And what's rather interesting is if you take the tumor cells out of the brain in this context and you grow them in culture and you treat them with the drug, you can see that they're equally sensitive to the tyrosine kinase inhibitor, similar IC50s. But if you retransplant them into the brain, you can reproducibly show this resistance phenotype that is preferential in the brain and that's enhanced by the tumor microenvironment. And again, if we apply our sequencing pipeline, what we see is that certainly while you can find gene signatures that differentiate these resistant cells from these sensitive cells, when they're grown in culture, you can see the number of genes and the magnitude of these gene responses increases significantly when these tumor cells are grown inside the brain. Again, suggesting that this in situ approach can capture some of these adaptive responses in the brain microenvironment. So I don't think I needed to show you all that for you for us to conclude that obviously the tumor microenvironment matters. So what I want to do for the rest of the talk is focus on a couple of more specific sets of findings. The first vignette will be for those of you that are lumpers, and so here what I'd like to show is some of the top-line analysis of this approach that we've used to look at gene responses in brain metastasis models that originate from multiple diseases, lung, breast, and melanoma. And then for the splitters, I'd like to also describe a little bit later an interesting phenomenon of lineage plasticity in the context of lung cancer and brain metastasis. So in this proof of principle experiment, what we decided to do was take 3 well-described models that are representative of triple-negative breast cancer, BRAF mutant melanoma, and KRAS P53 mutant lung cancers, in this case non-small cell lung cancer. And then established brain metastasis models from all 3, controlling for the location, in this case growing in the forebrain, and also overall tumor burden, and subject these to our BMX-seq pipeline. And in this case, we're looking first at stromal gene responses that are differentially induced in the stroma of tumor-bearing brain versus control brain, controlling for the region of the brain. And so what was somewhat surprising to me is that there was a lot of differences in the stromal response of these tumors. Again, even if we count for tumor burden and location, with the breast cancer model and lung cancer model showing the most significant overlap. However, if you focus on the core group of genes here that are commonly dysregulated in the stroma of these brain mets, what you find is a number of inflammatory molecules, perhaps not too surprisingly. And a lot of these are expressed in innate immune cells, but what was kind of intriguing to us was that we saw significant upregulation of 2 receptors in particular, LAG3 and HABCR2, also known as TIM3. And so for most of you in the audience, you'll probably are aware of the significance of these 2 receptors because they're generally thought as being expressed on lymphocytes and function as checkpoint receptors, and they're part of this new wave of checkpoint receptors that are being targeted for therapy. This was intriguing because this is, as you will recall, this was done in a xenograft model. These animals do not have T cells, and so why are these genes being expressed in the stroma of these brain metastases? And so if we look carefully, the reason is because we think that TIM3 and LAG3 are also expressed on brain metastasis-associated microglia. So this is immunostaining to confirm some of our bulk transcriptomic analysis, where you can see in yellow here, basically TIM3 and LAG3 overlapping with IBA1 positive tumor-associated macrophages. So in the brain, IBA1 can stain macrophages or myeloid cells that are either derived from the resident pool of microglia or also potentially from the bone marrow. We think that these cells are mostly, in these brain mets at least, derived from resident microglia because they co-stain with the microglial specific marker TME119, although not all of them are also TME119 positive. So this is also somewhat consistent with some observations that are made in the field of neurodegeneration and neuroinflammation, where some glial cell types and neurons can also express these receptors. Quite interesting. So we've also confirmed this in patient-derived xenograft models, and then more importantly, looking at the matched patient brain biopsy, we can also see consistent staining of TIM3 on IBA1 positive cells. And this is work done in collaboration with Veronica Chang and Katie Politi and several others, where we've started to amass a collection of patient-derived xenografts from brain metastasis patients. And then finally, we've confirmed this in syngeneic models, immunocompetent models of lung cancer as well. So if we turn now our attention to some of the gene responses from the tumors, from the same mice, same samples, we can see a little bit more overlap. And what we can do with some of this data now is integrate this with some of the stromal gene responses to kind of infer what would be the signaling pathways that are emanating from the brain microenvironment and that might be driving brain metastasis. And so what you see here, many of you will recognize immediately several components of the canonical Wnt pathway. And this is interesting for us because in the past, I'm not showing you the data here, it's been published, we had shown that this pathway is functionally required for brain metastasis in several models. But we were never certain whether or not the activation of this pathway was cell intrinsic or whether this was coming from the stroma. In this case, this model is predicting that a lot of these Wnt ligands or Wnt co-ligands are induced in the stroma of these brain metastases, leading to activation of the pathway in tumor cells. And so one prediction from this is that this pathway might be reversible. In fact, Emily did this very elegant experiment where she reproduced this finding by RNA sequencing, confirmed it by qPCR. I'm showing you here expression of AXIN2, which is a sensitive gene reporter of Wnt pathway activation. You can see as the cells are cultured in vitro or transplanted and grown as hindbrain mets or forebrain mets, Wnt pathway activation is induced. Then when you take a subset of these tumors, isolated tumor cells, and grow them back into culture, you can see that the pathway is shut down. So this is very reproducible and reversible. So the last thing I want to discuss with you is an interesting phenomenon of lineage plasticity in lung cancer. And this speaks to a context-dependent finding for lung adenocarcinomas in particular. And so here, I'm just showing you to the left here is a pathway analysis looking at some of the major gene responses that are induced in tumor cells, in this case, a non-small cell lung cancer model, lung adenocarcinoma to be specific, that is induced in the tumor cells when you compare them as they're grown in vitro or as flank tumors, also whether you compare them when they're grown in the lung for that matter. And what you see is that two of the most potently induced gene responses are genes that are typically associated with neuroendocrine tumors and also genes that are involved in axon guidance. And some of these genes here are confirmed by species-specific real-time PCR, including NCAM1, a neural cell adhesion molecule 1, which is a prototypical biomarker for neuroendocrine tumors of the lung. And we can validate some of these findings for these genes in PDX models. And so one subtlety that I want to point out is for some of these models, these cell line models, namely, for instance, this H2030BRM3 model, these are actually subpopulations of a prior parental population that's quite indolent and not capable of metastasizing to the brain. And so when we compare these metastatic cells in culture to their parental cell populations, again, these cells are a subpopulation of these parental cells. We can see that several of these neuroendocrine markers are actually already activated baseline in these cells, and that the expression is then further induced once these cells colonize the brain. So from prior experiments and our own experience, we know or we suspect that the differences between these cells at the genetic level, i.e. namely in terms of genetic mutations, there are very few genetic mutations, at least in coding genes, that differentiate this subpopulation from its more indolent counterpart. But rather, if we perform extensive epigenomic profiling, we can see that these cells are seemingly in a very different epigenetic state that also correlates with activation of some of these neuroendocrine genes. So here we're looking at ChIP-seq data, where you're looking here at ATAC-seq tracks. And just to walk you through, if you focus on this gene, for instance, here in the metastatic cell line, you can see that there's increased ATAC peaks, which are indicative of increased opening of chromatin regions. These regions, these valleys of open chromatin are nicely flanking peaks of increased histone H3K4 trimethylation, and this whole region of open chromatin and activation overlaps with the promoter of this gene FGF9, which tends to be overexpressed in neuroendocrine tumors. So we think what's interesting about these findings is that the cells that are, in this case, lung cancers that are capable or permissive for brain metastasis have a distinctive epigenetic state. And so in collaboration with Chin Yan and Trey Westbrook, we're just about finishing a forward genetic screen for chromatin modifying proteins that might be driving this. But rather than go into the mechanistic details of this, I want to take a step back and discuss what's the significance of this overall, or potential significance of this overall finding. And this has to do with considering different subtypes of lung cancers. So as you know, there are different subtypes of lung cancers that can arise in different areas of the brain. So for example, small cell lung cancers arise from the proximal airways, and it's believed that the cell of origin for these cells are in fact neuroendocrine cells, pulmonary neuroendocrine cells, whereas lung adenocarcinoma cells, which is representative of some of the models that I showed you, arise typically from alveolar cells in the distal airways. So although they have very distinct ontogenies, at least as demonstrated in experimental models, we also know clinically that some of these lung adenocarcinomas can manifest both morphological and proteomic markers of neuroendocrine features, And so these mixed neuroendocrine-like variants do exist in early stage resected tumors. A little bit of controversy as to what the clinical significance of these findings are. But then also what's quite interesting is that in the context of drug resistance, it's very clear that lung adenocarcinomas can convert to small cell lung cancer. And for the purposes of my discussion here, I think it's really interesting to note that in retrospective analysis of both retrospective data, but also autopsy data, small cell lung cancers, of all the lung cancers, has the highest predilection for metastasis to the brain. And so we ask a simple question. If we took the gene signature that is induced in these lung adenocarcinoma cells as they are exposed to the brain microenvironment and stratify primary lung adenocarcinomas, would it correlate with any interesting features? And in fact, what we find is that tumors that have high expression of this neuroendocrine signature, about 200 to 300 genes, have a higher propensity of developing or higher likelihood of relapsing in the brain. And so, and this is just a control to show you that many of the genes in this signature are in fact tend to be more highly expressed in neuroendocrine cells. So what are the biological implications of this? Well, the speculation, of course, is that we know from biology that neuroendocrine cells share many features with neurons, one of which is that they can coordinate neuronal inputs with secretory phenotypes. And so our current hypothesis is that for a subset of lung adenocarcinomas and perhaps other non-small cell lung cancers, that the appearance of these signatures is giving them a selective advantage for brain metastasis. Obviously, these cohorts are small, but we're interested. I'm really interested in revisiting this with additional studies. So speaking of biology, if we take some of these neuroendocrine-like genes and we knock them down in some of our models and inject them into mice, they do in fact attenuate the formation of metastasis. And some of these genes seem to be quite potent regulators of intracranial disease, reducing both the incidence but also the burden of brain metastatic progression. And so this is our current model. This is what we're thinking. We think that in a subset of tumors or subpopulation of tumor cells within a tumor in non-small cell lung cancers, they are in this epigenetic state that is permissive for the activation of neuroendocrine-like gene expression programs once they're in the brain, presumably due to some signals that are coming from the stroma. We have our obvious candidates based on some of the prior data that I showed you, looking at the stromal components and stromal changes in these metastasis models. So to summarize what I've shown you today, I think that just like everyone else, we're definitely interested in single-cell RNA sequencing approaches, and we have plans to integrate this with some of our analysis here. But I think the point is, even with a very simplistic bulk transcriptomic approach, we can infer some interesting findings and insights into the mechanisms of brain metastasis. And the chief advantage of this, I would argue, is that we really are not dissociating the cells from their host environment. And those single-cell technologies are very powerful. There are certainly some technical limitations that could introduce artifacts in some of these analyses. And we're particularly interested in deploying this in the context of drug resistance. So I'll finish with that and just mention that pending some temperamental editors and referees, we're looking forward to releasing a lot of this data, this rich data set through a web portal that will be accessible by anyone. So stay tuned and feel free to contact me. So with that, I'll take any questions that you might have.